Okay.
Okay. Oh, I think you need to enable screen sharing, Connie.
Yes, ma'am, I do. Okay.
All right, so if I do this, does it look correct?
Yes.
So it's to me, I see presenter mode.
How about this?
Yeah,
there you
that
go.
looks good.
Okay, so I want notes on my main screen. Okay, great. And then we've got a minute or two, Kyle or Carly, if you want to give it a go.
Yeah, sure, I can do
Still
that.
waiting for the other presenters.
Mm-hmm. Can you all see it?
Yeah, that looks great, Kyle.
Cool.
I can give mine a shot. See? Carly, we see your texts.
Oh. What about this?
Swap displays.
Is that
That
better?
looks good. Yeah.
All right.
It might be a matter of doing like a sharing just PowerPoint instead of the whole desktop. Okay. Well, I'm going to go ahead and share my screen as we get ramped up towards 4 o'clock here. Okay, great. All right, thanks everyone for being here. It's great to be back at the Beacon Congress and in an exciting session on with a lot of people who are thinking about antibiotic resistance in an evolutionary context. So today I'm going to talk about antibiotic resistance as it relates to phage resistance. And just quickly before I start, I just want to highlight all of these co-authors in blue have been my undergraduate research mentees on this project, and they've brought a lot of enthusiasm and a lot of great data over the years, and you'll see their faces throughout. Okay, so out there in nature, organisms are facing many, many diverse different selection pressures, including microbes. So they're under selection to grow fast, compete for resources, iron, to evade phage, to be antibiotic resistance, maintain osmotic balance, so on and so forth. So there's all of these different selection pressures, but often these pressures are encountered at the same time, but the evolutionary solutions might be opposite. So for example, you could imagine a population of cells like the one shown here. And these bacteria might be under selection by phage, which are gaining entry to the cell via structure. But that same structure might be used by the cell to efflux antibiotics from the cell. So the solution to the phage pressure might be to modify or get rid of this structure, but the solution to dealing with antibiotic resistance might be to upregulate or make more of this structure. And that could lead to a potential evolutionary trade-off between phage resistance and antibiotic resistance. And then understanding the form and prevalence of these trade-offs is important for a couple of reasons. One is just really important for understanding how diversity is generated and maintained in nature. And then two, there's a potential application to use phages to kill cells and then to sort of steer evolution to a state where antibiotic resistant structures might be downregulated and might be able to evolutionarily restore antibiotic sensitivity. So I'm really interested in these trade-offs, but first to test these trade-offs, we need a phage that would be tightly reliant on an antibiotic resistant structure like the one shown here. And one such structure is TolC. So TolC is an outer membrane component of an antibiotic efflux pump. So here you can see it's this red alpha beta barrel embedded in the outer membrane of a cell, and it works with these other components to efflux antibiotics out of the cell. But it's sitting here at the surface and has these surface exposed peptide loops, which could make it vulnerable to a phage. And we went out and found a phage called phage U136B that does use TolC as a receptor. I'm just going to quickly review a lot of work that we did on this for the sake of time, but it was identified via a screen of a global collection of E. coli phages collected by my colleague Ben Chan from around the world. And then we did a lot of work using molecular biology to show that this phage is really tightly reliant on TolC as a receptor. So it does not plaque at all on a TolC knockout. Plaquing of this phage is not inhibited at all by knocking out any other outer membrane protein receptors of E. coli. It does not grow on a TolC knockout in liquid culture. And then most recently, we showed that it does not absorb to a TolC knockout, and complementation of TolC restores absorption. So this phage is really tightly reliant on TolC. And so that brings us to our prediction, which is that phage U136B would select for TolC mutant antibiotic sensitive bacteria. So if we start with, say, an ancestral phenotype here that has TolC, that cell would be phage sensitive, but antibiotic resistant. And then after phage selection, we predict that we would see loss or modification of these protein pumps, resulting in phage resistant antibiotic sensitive cells. So to test this idea, I used two different methods. Method one was isolating pre-existing phage-resistant mutants. And so in this format of an experiment, sometimes called the fluctuation assay, started with wild-type cultures of E. coli. And in those wild-type cultures, there will be rare phage-resistant mutants sitting in there. And what we do is we just plate them on the surface of an LB agar plate with a copious amount of phage. And that phage selection will expose those pre-existing phage resistance mutations, and they'll be able to grow up into colonies that we can then isolate, confirm that they're phage resistant, and then go on to test if their antibiotic resistance is also changed. The second method is using experimental evolution. So it's a similar setup, except that in this case, the bacteria are serially passaged with their phages for a period of time. And so mutations are allowed to grow and compete with one another in this community context. So here competition really matters. And so we might see different endpoints in terms of which phage mutations are actually evolutionarily successful. So first I started with method one, and starting with 20 independent cultures of E. coli, isolated 20 independent phage resistant isolates, confirmed that they were indeed phage resistant, and then tested their sensitivity to an antibiotic known to be effluxed by TolC, which is tetracycline. So what I'm showing you here in this plot is tetracycline resistance indicated by the minimum inhibitory concentration, or the MIC, along the x-axis, increasing resistance from left to right, and then the number of mutants out of 20 along the y-axis. What you can see here is this is where the ancestral E. coli starts in terms of tetracycline resistance. And then among these phage-resistant mutants, we do see this group of mutants right here, which have reduced tetracycline resistance. I also sequenced their genomes and confirmed that these mutants do have mutations in TolC. So these mutants over here are very consistent with this evolutionary trade-off idea and what we predicted in terms of phage resistance and antibiotic resistance. However, what really surprised me were these mutants over here, which actually have increased tetracycline resistance. So even though they're selected by this phage, which really needs TolC, somehow the phage resistant mutants are actually more resistant to tetracycline. So this was puzzling, and we did a bunch of work to figure it out. And it turns out that these are LPS synthesis mutants. LPS, lipopolysaccharide, is important because it provides a protective barrier to the cell, and it's highly involved in antibiotic permeability. And lipopolysaccharide is also a common receptor for many phages. About half of the E. coli phages that have been surveyed actually rely on LPS, and U136B is no different. So we did a lot of molecular work knocking out various LPS synthesis genes, complementing that and showing that phage U136B relies on this. So because of that, the bacteria can easily find a way around this trade-off by evolving LPS changes through LPS, which affects phage entry into the cell, and then also increases the effectiveness of the barrier to tetracycline. So that was pretty surprising. And then we also found that these mutant LPS molecules, they have different effects on different antibiotics. Here with tetracycline, we saw that some of these mutants had increased tetracycline resistance. When we look at this other antibiotic, colistin, we actually see the opposite effect. The ancestral MIC of colistin is right here, and many of these mutants have reduced colistin resistance. And this is because colistin is an antibiotic that interacts with the cell membrane. And when the cell has these mutant LPS conformations, that makes it actually easier for colistin to get in there and to disrupt the cell. So here we have sort of a trade-off, but it depends a lot on which specific antibiotic as well as which specific phage resistance mutation is in that cell. Okay, so that was all with method 1, just looking at which mutations are there, sort of a survey of all the possible mutations. Then with method 2, I looked at what happens after a period of experimental evolution. And so I conducted just a small evolution experiment, 10 days, serial transfers in the same medium. We had 10 populations with phage present. The phage were allowed to grow and replicate. They weren't replenished each day. And we had 10 control populations with no phage. And there were no antibiotics present in any of these populations. After 10 days, isolated bacteria from each of the populations and confirmed phage resistance, and then went ahead and looked at tetracycline resistance and colistin resistance. So here is just the summary from the mutant isolation that I showed earlier, where 30% of the isolates had TolC mutations. After experimental evolution, we now see quite a different result where 100% of the isolates have TolC mutations. And what's interesting is that because of that, we no longer see this signal of a trade up between phage resistance and tetracycline resistance. We see more of a trade off with tetracycline resistance. However, I do really want to point out that there are still quite a few mutants in here that are phage resistant and they have TolC mutations, but they maintain their wild type tetracycline resistance levels. And then likewise with colistin, we no longer see such an effect of the trade-off. And again, that's because we're not seeing these lipopolysaccharide resistance mutations. Okay, so overall, what we've seen here is that changes to antibiotic sensitivity depend on a few things: the specific phage resistance mutation, the antibiotic that we're using, and then the type of selection experiment, whether it's a traditional selection experiment or an evolution experiment. So all of those results led me to think, well, how generalizable are these results? Or have we just kind of characterized, some case examples of particular antibiotics with this one phage? And so what I'm going to present next are some very new data where we looked across 6 additional antibiotics from 6 different antibiotic classes with these same mutants from the traditional selection experiments and the evolution experiments. Okay. So what I have here are the 6 additional antibiotics across the x-axis and then relative resistance along the y-axis. If there's no change to the MIC of the antibiotic, it'll appear right here. If it's more resistant to the antibiotic, it will appear above the line. And if it's less resistant to the antibiotic, it will appear below the line. And first I'll show the results for these, the traditional mutant selection isolates. And here are the results. And what you can see is that there is a general pattern of a trade-off. These phage-resistant isolates do tend to have reduced resistance to a wide variety of these antibiotics, but it really depends a lot on the specific antibiotic as well as the specific mutation. So you can see that among the individual points, those are the individual isolates. There are many that might be slightly more resistant to the antibiotic, and there are plenty right along the 1-to-1 line indicating that they've either evaded the trade-up or the trade-off, they don't pay any cost to resistance, or we're unable to detect a difference. And then here are the results from the experimentally evolved isolates. Those are shown in pink here. These were evolved in the presence of the bacteriophage, and the gray bars are the corresponding controls in the absence of phage, again, for each of the 6 antibiotics. And what you can see here is, again, a pattern where we see a lot of trade-offs where the phage-resistant mutants are more resistant to, are less resistant to the antibiotic. But again, there's a lot of variation depending on the specific mutations and among the different antibiotics. So just the overall summary, phage D136B needs both TolC and lipopolysaccharide. Trade-offs between phage resistance and antibiotic resistance appear more common and are stronger than trade-offs, but trade-offs and trade-off evasion is still pretty common. And then changes to antibiotic sensitivity depend on the specific phage resistance mutation, the antibiotic, and the type of selection experiment. So I'll leave the slide up here. Carli's gonna speak a little bit later in the session about the effect that host genetic background can have on these patterns. And then Eddie and Yvonne are going to be talking about what's happening on the phage side of things, because it turns out that the phage are evolving throughout this entire process as well. And we have some interesting results from that. Okay, so I want to thank all of my undergrads that have worked with me over the years. Funding Beacon has been great and really a great source of inspiration, ideas, as well as funding over the years. And of course, the Turner Lab and all of my other students. So yes, if anybody has questions, feel free to just jump in and unmute and ask. Otherwise, I'll try to monitor chat. And then in the meantime, I think Kyle, you're next. You can start getting set up.
I actually have a question. Wonderful talk. This research is awesome. I have actually two questions, one specific to your study and one more general question. So firstly, have you looked at how resistant the E. coli has become to the phage itself and whether that correlates with the magnitude of the trade-ups or the trade-offs?
Yes, we have. In the evolution experiment, they're pretty much completely resistant to the ancestor. And then among the fluctuation assay mutants, they vary a little bit. I'm gonna let Carli talk about that because I think she's got a slide or two, but we can definitely come back to that.
Cool. And my one general question is, so actually this kind of harkens back to the discussion after Chris Waters' talk yesterday. So viruses are such a large selective pressure for bacteria, right? Because by mass, they greatly outweigh any sort of organism on Earth. Viruses are innumerable. So given that, what do you think the prevalence is for trade-offs or trade-ups among bacteria in general to phage that they encounter in the environment?
Great question. I've thought about this. I'm going to cut myself off though because it's your time to present, and I'll try to address that in the chat.
Cool. Okay.
Or we can chat later. Thanks, Kyle.
Awesome. Okay, so for those who don't know me, my name is Kyle Card. I'm a postdoc in Jake Scott's lab at the Cleveland Clinic. And I will be talking about some work that we've been doing centered around the multifaceted nature of antibiotic resistance evolution. So the evolutionary potential of resistance, it may depend upon several factors. So as a graduate student in Rich's lab, I found that the genetic background of an organism can affect both the phenotypic and genotypic evolution of antibiotic resistance. But we also know that population size and mutation rate parameters may affect this phenomenon as well. So to dive more deep into this, so population size can influence resistance evolution. So let's imagine that we have two populations of different sizes, and let's assume that they have the same point mutation rate. So given that the mutation supply rate is proportional to the product of the population size and mutation rate, we would expect that on balance, large populations should be more evolvable because they are more likely to sample rare mutations that provide high levels of resistance. Nonetheless, there are several confounding effects that might impinge upon this expectation. So these two populations might have potential differences in mutation rates, or they might have potential differences in genetic background. So I'm particularly interested in this related question. How do population size, mutation rate, and genetic background interact to influence evolutionary paths to resistance? So to address this question, I teamed up with fellow postdoc in the lab, Jeff Baltus, to sort of approach this question from a computational perspective. And just as a point of note, so I'm going to show some following schematics, and they are the simplified illustrations of the underlying evolution that is occurring in our model. So what we do is we first evolve populations from different initial genotypes with no selection. So we simulate fitness landscapes where every genotype on the landscape has equivalent fitness. So in effect, the populations under this initial phase of our simulations are evolving under genetic drift. So I'm showing two represented populations, A and B here. And we evolve these populations in our simulations for 500 generations, or 500 updates. Then we generate a drug landscape. So in contrast to the landscape I just showed in the previous slide, this landscape has some topography to it. So it has areas where genotypes correspond to lower fitness and genotypes that correspond to higher fitness. And the degree of ruggedness of the landscape is actually tuned in our model through an epistasis parameter. And then we take the offspring that evolved under the neutral landscape, under drift, and we introduce them, we just plot them into the drug landscape and we measure evolvability. So in our study, evolvability corresponds to the fitness difference between an individual's final genotype and the population's initial genotype. So we focus our analysis on the most evolvable individuals from each population. So for example, in this schematic illustration, population A at the top, it starts at a fitness value of 4, and a descendant of this population found a global optimum at fitness value of 7. The difference is 3. And likewise, for population B, the evolvability is 4. So this is how we're defining evolvability in a study. So now we decide to build an intuition that by altering the population size and mutation rate parameters, these may lead to different outcomes. So let's imagine we set the simulations with large population sizes and or high mutation rates. In this case, population A, it starts close to the global optimum under the neutral evolution phase. And then when we plot it into the drug landscape, a descendant actually inhabits the global optimum. And however, under small population sizes and or low mutation rates, there's less sequence-based exploration of these populations. In this case, population A did not find the optimum. And on the other hand, offspring for population B found it reached a higher resistance level compared to offspring from population A. So we can see that not only population size and mutation rate affect evolvability, but also the starting location, which is history, can also affect this phenomenon as well. So here I'm showing simulation data. So we found that the average evolvability in our simulations increases of population size and mutation rate. So here I'm showing data on fitness landscapes for 32 initial genotypes. So on the x-axis, I'm showing 5 different mutation rates, and on the y-axis, I'm showing 5 different population sizes. So we see in the lower right-hand corner of these heat maps, highest evolvability. And these results sort of are what we expect given what we know about mutational supply rates. We also see this trend over fitness landscapes of increasing size from 32 initial genotypes to 1,024. In a separate experiment, we evolved populations from different starting genotypes as before. And then we find the maximum and the minimum evolvability among these different genotypes. So again, the schematic illustration, there are only 2 populations that I'm representing. What we do is we subtract these values, these evolvability values, to determine the maximum effect that starting genotype has on evolvability. In other words, this is looking at the effect of history. So when landscapes are small, history's influence is most pronounced at high mutation rates and large population sizes, shown here in the heat map, where the maximum effect due to studying genotype or history can be seen in the lower right-hand corner of the heat map. But as genotypic space increases, history's influence becomes more pronounced when population sizes are small. So this trend across increasing fitness landscapes actually shifts to the upper right-hand corner of the heat map. So why is this the case? So why does history's influence on evolvability change with increasing genotypic space? Okay, so we can sort of now imagine this genotypic space with 1,024 genotypes. And here I'm showing two populations, A and B. So these are just idealized schematics of such populations. The initial genotype, so where the population starts off in under sort of, before that initial phase of mutual evolution, is shown as these gray circles on the landscape. And the offspring of the population is shown as these teal circles on the landscape. So when a population with 10 individuals, when we have a population with 10 individuals, they will explore on average about 1% of all possible genotypes on this landscape after this phase of evolution, such that differences in evolvability should therefore be more pronounced in small populations because individuals may end up at more extreme fitness values just due to chance. So this work and future work in both bacteria and cancer sort of get at this broader question of public health relevance, and that is how predictable is resistance evolution. Because if we can better understand the factors that underlie evolvability and resistance evolution in general, but more specifically, we might be better equipped to treat resistant populations of both bacteria and cancer through rational treatment strategies. So we hope that our work is a stepping stone in this direction. So with that, I want to make a couple of acknowledgements. My postdoc advisor, Jacob Scott, my collaborator, Jeff Maltus. I want to thank Theory Division as a whole and HHMI for a Henry Gray Fellowship. And I'd be happy to take any questions.
Great, thanks, Kyle. If people have questions, you can type them into chat or just unmute and ask them. There's a question in chat. Have you looked at landscapes with varying levels of ruggedness to see how that impacts the evolution?
We have. Is it okay if I share my screen? I could show some quick results. I'm sorry. Oh, no worries.
Oh, no problem.
We have, Devin. So we actually, like I said, we can tune the epistasis in our model, which affects the ruggedness of the landscapes. So we focused our analysis on smaller landscapes just for computational simplicity. But we found that actually the trend is relatively similar between landscapes with moderate levels of epistasis and no epistasis at all. But actually the effect of history is more pronounced when epistasis actually is there. So unfortunately, I probably don't have time to go into too much detail, but I would love to either chat and talk about in the chat or maybe over beers tonight in Bradford Town.
there are other questions, you can jump in. We have another minute or two. A lot of accolades in the text, Kyle. You can check out as you have time. Oh, Chris, go ahead.
Yeah, I got a question. Good to see you, Kyle. Thanks for the talk. So in your simulations, there's no real trade-off for a high mutation rate, right? So, what do you think about if you factored that in? How would your results be impacted?
So trade-off being, so I guess what do you mean by trade-off with high mutation?
Well, I mean, as bacteria have very high mutation rates, then they start
more deleterious mutations.
Yeah, deleterious mutations.
Yeah, that's a good question. It's a good question, actually, in theory. So one can imagine that you actually start close to an optimum even in neutral space. But if you have a high mutation rate, you actually are sort of going further away from that optimum. So actually, yeah, that's an important point. There's in some sense a trade-off. With more mutation rate, you actually can get further away from where you actually ought to be and need to be. That's most
right. And then there's multiple optimums too, right?
Exactly. Yeah, it depends upon the landscape, but yeah, there are definitely multiple optimums as well. So it can get pretty tricky and dicey.
All right, cool. Thanks.
Yep.
Thanks, Kyle.
Thank you, guys.
All right. Am I ready now?
Yep, looks good.
Okay. All right. So good afternoon, everyone. My name is Mizpha Fernander, and I will be doing my Beacon meeting talk on evaluating the genotypic and phenotypic changes in Streptococcus mutans adaptations to microgravity using experimental evolution. So first, by 2025 and 2033, NASA is developing capabilities to send humans to an asteroid and Mars explorations. Although microbes are not only ubiquitous on Earth, but they are in space as well, and the crew themselves is the primary contaminating factor. As a result, infection of unknown bacteria is not necessarily a primary concern. However, the infections resulting from the normal flora of the crew are. When looking at dental infections in space, based on the integrated mathematical model, it predicts that dental emergencies will be one of the top 5 conditions impacting future space missions, as seen here by this table from the NASA Dental Summit in 2012, with caries having 39% of incidents. Also, long-term space missions and increased exposure to radiation and microgravity leads to an increased incidence of oral disease in astronauts. So when seeing or observing the karyogenicity of S mutans, when present in high concentrations, S mutans adheres to the tooth enamel as a biofilm and causes cavities, as seen here by the electron microscopic images in its both planktonic form and biofilm form. Other characteristics include it being gram-positive, a facultative anaerobe, and commonly found in the oral cavity. Essentially, there are 3 main ways to assess virulence of Streptococcus mutans. That includes oxidative stress tolerance, which looks at the ability to assess the early colonizers of hydrogen peroxide, of S. gordoni and Streptococcus sanguinis. Also, its ability to adhere and join biofilm, viewed here, and determine the ability to tolerate high levels of acid produced during fermentation, seen here on the schematic. For the research objective is to examine the adaptive response of S mutans as a result of long-term space travel. In order to conduct microgravity on Earth-based experiments, we use high aspect ratio vessels, also known as HARVs, to assess normal gravity and microgravity. When examining microgravity, it is parallel to the axis of rotation. And basically from the schematic, it just shows that in microgravity, it simulates bacteria being in constant free fall and never settling to the bottom. in normal gravity, seen also in the schematic and image, is perpendicular to the angle of rotation, and it acts in only one direction where all of the bacteria settles at the bottom. In order to look at the long-term adaptation and experimental evolution, you take the ancestral population and subculture it into 4 control populations representing normal gravity, as well as 4 treatment populations representing microgravity. From there, we're able to test for contamination twice a week and store cultures in case of contamination via a glycerol stock. Subculture conditions occur every 36 to 48 hours over the course of 100 days, where phenotypic assays are assessed every 3 weeks, including adherence, acid tolerance, oxidative stress tolerance, as well as antibiotic resistance assays, or MICs. Genomic assessment is also observed, where we do whole genome DNA sequencing for 3-week populations, as well as RNA sequencing for 100-day cultures. When looking at the mutations that are unique to both normal and microgravity, we found that there are very few mutations detected that were unique to only microgravity populations. However, at 9 weeks, we detected 3 specific mutations seen here as CCPA, ENO, and YLQF. At 100 days, there were 8 unique mutations identified here, and we also detected them in a single population as well. And I'll also discuss some more mutations. So in mutations specific to microgravity and normal gravity as well, 7 mutations were initially identified in normal gravity and later identified in microgravity populations listed here. There were 4 mutations identified in microgravity populations and later in normal gravity populations. And 2 mutations were detected in both populations listed here. Moreover, 1 mutation was detected at 100 days for the first time in both populations, known as LYTR, which I will discuss later. So looking at sucrose-dependent adhesions, we saw no mutations in any of these genes for normal gravity 2 through 4 populations. And there was mutations in the K that interact with glucotransferases as well as glucan binding proteins in sucrose-dependent adhesion, dependent adhesion, seen here by this tooth schematic. We also identified a reduction in adhesion in 2 normal gravity populations as well as 3 microgravity populations. For sucrose-independent adhesion, all populations had the SMU399 mutation, which interacts with the COMX or operon regulator system involved in sucrose-independent adhesion and mutations that activate SPA-P, seen here by the ANG1 and 2 SPA-P for the tooth schematic. MG4 and MG3 were the only populations in which the SMU_399 mutation had not undergone fixation. Moreover, MG4 and MG3 held the S721I mutation in SPA-P. In acid tolerance that was reduced due to microgravity, overall, the normal gravity populations behave very similar to the ancestral population, with almost complete reduction in colony forming units by 30 minutes of acid exposure. But more specifically, in the microgravity populations, MG1 had a PMPA protein that was activated and interacted directly with the RPOE mutation. Also in MG3, the LYTR mutation, which I discussed earlier, decreased acid tolerance, having the T243N mutation. in microgravity and oxidative stress susceptibility, as shown on the left with the Streptococcus sanguinis and Streptococcus mutans ancestral populations, there's very little death resulting when S. sanguinis is grown next to the ancestral population seen here. However, when grown in microgravity populations identified for the remaining images on the slide, there is increased death and therefore increased susceptibility to the hydrogen peroxide produced by Streptococcus sanguinis, seen here with this being the Streptococcus sanguinis and this being the Streptococcus mutans. In summary, for our genotypes, microgravity is a weakly selecting environment, and microgravity adaptation experiments do not consider normal gravity as an adaptive environment with multiple replicants and sequencing at multiple time points. But we cannot conclude specific adaptive responses of S. mutans to microgravity. However, we can conclude the importance of having multiple replicates and time points for weakly selecting microgravity environments. In our phenotypic summary, there was no change in sucrose-dependent or independent adhesion specific to microgravity. However, there was increased susceptibility to both acid and oxidative stress as a result of microgravity environments. Lastly, I would like to acknowledge Dr. Misty Thomas and my lab team, both my former and current lab colleagues. I would like to also acknowledge Dr. Joseph L. Graves, who helped with the genotypic data sequencing, as well as the Beacon funding. And I would like to acknowledge my RISE Fellowship Scholar and the coordinators, Ms. Anita Sanders, as well as Dr. Keith Schimmel. Thank you. This is my contact information. Any questions?
Great, thanks, Mizpha.
All right.
So people can jump in with questions or type them into chat. I just had one quick question. In your experiments where you can compare all of these phenotypes after normal and microgravity, were those on populations or were they on isolates of mutants from the populations?
What do you mean? Like with the...
Like, did you take the entire evolved population, like a sample right from the microgravity chamber, or did you isolate individual bacteria from, were they representative isolates or individuals from the population?
Well, we subcultured like a population like for over the course of the 100 days. So we would subculture like a sample, like a bacterial culture sample for the 100 days, and would do the phenotypic assay. The phenotypic assay is for every 3 weeks from that population that we collected.
Got it. And then there's a question from Ben Kerr, stellar work, Mizpha. I'm curious about the possibility of trade-offs in your system for mutations that are specific to microgravity. Have you performed competition experiments between these mutants and their ancestors in the normal gravity environment? For example, are the MG mutants selected against a normal gravity? Thanks again for an interesting talk.
No problem. So have I done like a competition assay?
Mm-hmm.
I've done an assay similar to a competition assay, but not exactly a competition assay. But in the upcoming assays I'm doing for my co-adaptation to microgravity and silver, I will actually do more full-on competition assays.
And then
but that's coming up this semester.
Great. And then a question from Nkrumah. Have you considered doing experiments for both microgravity and radiation or factors? Imagine latent phage becoming lytic, which was, of course, which is of course another thing to consider when treating astronauts. Great talk.
Thank you. Radiation factors. No, I have not considered that, but that is something to look into though.
Yeah, and also if I can get a quick one in, I think that you guys are dramatically understating how much adaptation is going on to microgravity. And I know I've talked to you guys about that. And that's because the way you're determining that mutations are unique is whether they only appear in one gene. And when I looked at the data, there are mutations that occur only in microgravity, even if they are appearing in the same gene as mutations that occur in normal gravity, but these mutations are never the same. And there are reasons to believe that how proteins fold in various gravity regimes may be important adaptive aspects of what happens. And so, in fact, one quick thing is that a lot of those mutations that were found, or unique microgravity mutations that were found, were the ones that swept and that had frequencies of 0.9, 1. Mm-hmm. But then a different mutation in normal gravity was at a different frequency. So I actually think you guys have much more adaptation in microgravity than you're reporting. I think you're being rather conservative.
Thank you very much, Dr. Graves.
Kyle, did you have a question?
Oh, no.
Just stretching. Okay, great. Let's thank Mizpha again.
Thank you for listening.
All right, that's great.
Hello, everyone. I'm Olusola Jeje. And I'll be speaking on environmental evolution of E. coli K12 when exposed to excess iron 3 and T7 bacteriophage. And I'll be looking at the consequences of antimicrobial substances. Evolutionary trade-off is actually the background basis for this research work. The diagram to the right here shows organisms that have been exposed to traits 2 for organism X5 is actually responding to acid, while X1 is being grown rapidly on lactose. And for X1 to gain some ability to resist acid, then it has to kind of lose some ability to grow rapidly and move down the curve. Whereas for X5 to gain some ability to grow rapidly, it has to lose some resistance to acidity and moves down to probably X4 or X3. So for nature to spend on one side, definitely it has to lose on the other side. So that's the principle of trade-off. And like I've said, that's what this research is actually, the background of my research. Iron is actually very important to bacteria. And reports shows that without iron, bacteria may not really be able to reproduce. And along the line to ionic and nanoparticle methods have been proposed as antimicrobial. They have properties to kind of inhibit the growth or kill bacteria altogether. And this includes silver, we have copper, we have zinc, we have SS iron 2, 3, and gallium. But then along the line, it has been reported that eventually the bacteria can become resistant to excess of this, material, in which case, the materials are unable to actually kill the bacteria again. Then bacteriophage 2 has been proposed as good alternative to actually take care of multi-drug resistant bacteria. And they have some properties in the sense that they are not detrimental to the human host. Then they could self-replicate at the site of infection. Then reports also shows that we have resistance to phages being developed along the line as well. So what I will now be looking at is there a trade-off in E. coli between iron 3 resistance and T7 phage resistance? What I did was to create variants of iron 3 selected. The set of bacteria that were exposed to SS iron 3. Then I have another population in which were exposed to SS iron 3 and phage. Then I have another population that was exposed only to phage. Then I determine the phenotypic changes, the growth curves, how they behave in those medium. Then I determine the genotype as well to know if there's anything that changed in the genes of the bacteria. This is like the template of the experiment. Like I said earlier, I have 4 populations, ion 3, phage selected, I have ion 3 selected, I have phage selected, and I have a control population. Now they are suspended in 10 ml of DMB media, and I do daily transfer. Then after that, I conduct MIC on them to determine how they behave in SX, ion 3, ion 2, silver, gallium. Then after that, I determine how they grow as well in tetracycline and ampicillin, rifampicin, and 2 other antibiotics. Yeah, these are part of the setup of the experiments. Now, the results shows that now this graph is optical density. So these are concentrations of the ion 3 that were tested. And this graph, the purple ones are ion 3 and phage selected, while the blue is ion 3 selected, that's ion 3 only. Then I have the control as red, then I have the phage selected as a green, then the ancestor as the black dots there. This shows that ion 3 selected are actually doing better than every other population in this. And this was actually significant when tested. The F value was 10.88 here. Then the next one I'll be showing is they were actually tested also in gallium, the silver nitrate, and ion 2. This is a graph for gallium. And here it shows that both ion 3 selected and ion 3 phage selected were not actually too different in the way they behave, whereas both of them actually had superior growth when compared to the control, the phage selected, and the ancestor. Then also in ion 2, the pattern in ion 2 also shows that ion 3 selected and phage selected and ion 3 selected are doing far better compared to all other 3 populations. And lastly, in silver, ion 3 and phage selected, that's a population exposed to both, were actually better in terms of growth compared to ion 3 selected, the control, the phage selected, and the ancestor. And what I did next was to probably see after being exposed to SS ion 3, what happened when they are tested against SS antibiotics. And I tested ampicillin, tetracycline, sulfonamide, chlorphenicol, and rifampicin. And the result shows this is ampicillin. I took optical density after 24 hours of exposure to SS of these at these concentrations. And what it shows is that my ion 3 and phage selected were actually better compared to ion 3 selected and other 3 populations. And this was also significant when tested statistically. The next one is when exposed to tetracycline, and it shows as well that the ion 3 selected and the ion 3 populations doing better. Then we have the phage selected as well coming up, while the control and the ancestor were not actually doing better compared to the other 3 populations. Then I have sulfonamide. The trend 2 was almost the same. We have both the ion 3 and the ion 3 phage selected doing better compared to the 3 other populations. In chloramphenicol, the trend 2 is not different. We have the ion 3 selected and the phage selected coming out compared to the 3 other populations. So in summary on that, it shows that the when they were actually exposed to SS ion 3 and phage T7, then they behave like they have superior growth in SS antibiotics compared to other populations that were not exposed. And then I will now be asking the next question: does ion and phage selection confer any genetic change on E. coli? Now, what I did next was to do full DNA sequencing for the ion 3 phage selected population, for the phage selected population, then the ion 3 selected population only. And the result shows we actually have the genes like the ARPO-C, ARPO-B that were regular genes that bacteria used to kind of, in response to stress. But then in phage selected populations, I have the WAC gene, which was actually response to envelope stress, which was induced by the phage. And interestingly, this was found only in the phage selected population. Then it was actually found in the phage and ion 3 selected population as well, to show that it's actually a response to phage only. Then ion 3 selected shows genes that shows that's bacterial display when they're actually under stress, which might be nutrients or some other factors, or when they are exposed to excess of those. What the study we found out in the study was that there was no trade-off between ion 3 and ion 3 phage selected E. coli. They were resistant to ion 3, to silver, to nitrate, to ion 2 and silver nitrate, but we couldn't really find any evidence that shows trade-off between the two. Then ion 3 and ion 2 phage selected E. coli were resistant to ampicillin, sulfonamide, chloramphenicol, and tetracycline. Then ion 3 phage selected enhances antimicrobial resistance. Like I showed earlier, the ones that were exposed to ion 3 and phage had the ability to resist conventional antibiotics compared to other populations that were not exposed. Then, contrary to expectations, selection in ion 3 and T7 phage seem to have some synergistic effect, but because in some cases, we discover like when they were exposed to ion 3 before phage, then they don't really need to involve any specific mutation to resist phage. Again, they actually use the same resistance, the same mutation that were developed to ion 3 to fight phage. Then when E. coli cells are exposed to various stress conditions, general stress responses were actually induced, which was evident in almost all the populations when they were sequenced, like the ARPO-X. The WAC mutation, like I've said earlier, which was response to envelope stress, was only found in the population that were exposed to Escherichia phage T7, to show that they were actually responding to envelope stress. Then ion 3 selected populations, like I've said earlier, did not involve WAC, suggesting that probably some Pleiotropy is actually playing here, in which the genes that were used to resist excess ion 3 were being used again against the phage. So that was the summary. I would like to acknowledge Beacon, Tricep, my PI, Dr. Graves, and my colleagues in the laboratory for the part they play in the success of the research. Thank you. Any question?
Great, thanks, Olusola. It's really interesting that your results kind of converge with ours, where you see phage resistance that can then also result in increased antibiotic resistance. And I wonder how much of that is driven by LPS mutations. I noticed you had WAC mutations, and that's one of the determinants, I think, of the inner core of the lipopolysaccharide, similar to what we've seen. So it's interesting to see multiple studies coming up with these similar results.
Thank you.
The question from chat, do you know if the mutations in RPOB selected in iron 3 occurred in known hotspots of that gene that confer direct resistance to rifampicin?
I didn't hear the question well. I didn't hear the question well.
I can ask it correctly if you want to.
Yeah, go ahead.
Okay. Okay.
Oh, I just was interested in so the rifampicin binds to the beta subunit of RNA polymerase. So
okay.
There's known hotspots in that gene. Like I think there's 2 clusters of known hotspots where you get direct resistance to rifampicin because it presumably disrupts binding. So I'm just curious where those mutations, whether those mutations happen to land in some of those hotspots or if it's something else that's going on.
Well, I think from with the response we have to other antibiotics, we want to believe like it might actually be the ability of the bacteria to resist antibiotics when they were exposed to iron. And actually the polymerase for T7 actually has a spot for rifampicin resistance. So I wouldn't know if that one might actually play any role there, but then we believe like the same response that we have to other antibiotics is what is actually taking place with rifampicin as well.
Yeah, and also Solo, it's easy enough to check because we have done experiments where that's exactly what happened, where we had mutations in RPOB that were in the hotspots and those populations were resistant to rifampicin. So I think Dr. Kerr is simply asking you or suggesting that you check to see if those mutations are in the places that usually confer resistance to rifampicin.
Okay.
Yeah, there's a nice paper by Mary Reynolds I can send you the reference by Mary Reynolds in 2000, which goes through where those clusters are.
Okay. Thank you.
All right, thanks again.
Hi, everyone. Can you see my screen okay?
Looks good.
All right. So my name is Carli Roush. I'm an undergraduate senior at Yale University, and my project is investigating relationships between phage resistance and antibiotic sensitivity in uropathogenic and laboratory strains of E. coli. And this was conducted through the Turner Lab in conjunction with Alita Burmeister. So for some background on this project, antibiotic resistance in persistent bacterial infections is a large and growing problem in the medical field. And bacteriophage, which are viruses that selectively infect bacteria, can and have been used as potential alternative treatments for these infections. So phage U136B, which is the phage I'm specifically talking about today, was isolated from feces on a pig farm in Connecticut, USA. And it's shown in this transmission electron microscope image. U136B uses the outer membrane protein TolC as a phage receptor, and TolC is the main antibiotic efflux pump in E. coli bacteria. This is important because it can lead to a sort of trade-off. So if the TolC is present and functional, then the antibiotics are effluxed out of the cell before they can kill the bacteria. However, bacteriophage can readily bind to and infect the bacteria. So a trade-off shown below is the ideal situation in terms of phage therapy. So as discussed, here the bacteria is antibiotic resistant but phage sensitive. However, after a TolC mutation where the TolC is missing or mutated, the phage is no longer able to bind, so the bacteria is phage resistant. However, the antibiotics are not able to be effluxed, so the bacteria is antibiotic sensitive. So this is a trade-off between antibiotic resistance and phage sensitivity. However, trade-ups between these two also exist, as discussed by Alita Burmeister in her talk a few minutes ago. And she demonstrated that there are both trade-offs and trade-ups found in the E. coli laboratory strain K12 for antibiotics from different classes. And a little bit about host genetic background. So E. coli laboratory strains K12 and B are commonly used non-pathogenic model organisms that came from different lineages of E. coli. And our uropathogenic E. coli, or UPEC strain that I'll be talking about, is a pathogen which was initially isolated from a human patient with an acute kidney infection. And it has been previously found that phage E136B has different infectivity levels on each of these hosts. So the research questions that I sought to answer were, does host genetic background influence the relationship between resistance to phage E136B and sensitivity to antibiotics? And is there a difference in antibiotic sensitivity between E. coli mutants, which are partially resistant to phage E136B, and those that are fully phage resistant? So for an overview on some methods, isolating the phage resistant mutants, so each bacterial host was mixed with phage E136B. This mixture was plated and incubated overnight, and then any bacterial colonies which were able to grow in the presence of phage were isolated as phage-resistant mutants of the original host strain, which was E. coli B or UPEC. And so 10 mutants from each host were isolated. The methods for measuring phage resistance included diluting phage E136B, mixing it with host bacteria, plating and incubating overnight, and then plaques were counted for each bacterial mutant and compared to the wild type strain to obtain the efficiency of plaquing. And plaque is this clearing shown here on this plate where phages were able to bind to, infect, and kill the bacterial cells. Finally, my methods for measuring antibiotic sensitivity. I used MIC test strips. So here bacteria was spread on a plate and an antibiotic test strip was placed in the middle of the plate and incubated overnight. Then the minimum inhibitory concentration, or the MIC, was recorded at the lowest point where bacteria could not grow. So you can see in this image, this zone of inhibition around the bacterial strip, and the MIC would be recorded at the point where the zone of inhibition begins. So moving on to the results, for the E. coli B strain mutants, we found that all 10 mutants were confirmed to be fully phage resistant, and we found that antibiotic sensitivity varies by the antibiotic. So this figure is showing the efficiency of plaquing that I mentioned earlier. On the x-axis is each individual bacterial mutant, and on the y-axis is a measure of efficiency of plaquing compared to the wild type. So a level of 1 would mean no change from the wild type. Below 1 would be a decrease in infectivity, and above 1 would be an increase in infectivity. And so you can see for each of these 10 mutants, they have a value below the limit of detection. So we were not able to see plaquing for any of these strains. And so these were determined to be completely phage resistant. And here are the results of the MIC tests. So again, the mutants are on the x-axis, and then the fold MIC change compared to the wild type bacteria is shown on the y-axis. So similarly, a level, a value of 1 indicates no change from the wild type bacteria. A value above 1 indicates greater resistance to an antibiotic, while a value below 1 indicates greater sensitivity. So you can see that the effects vary by antibiotic. For example, in trimethoprim sulfamethoxazole, you can see that there's a trade-off. And so all of these mutants are phage resistant, and you can see that they're also below the level of 1, so they have an increase in sensitivity. However, in an antibiotic like ciprofloxacin, we see a trade up. So these phage resistant mutants are also more resistant to this antibiotic. And for oxacillin, it may look like there's no change, but this is just due to the fact that all of these levels were above the limit of detection for this particular method. So we actually have no way of determining at this point whether there are trade-offs or trade-ups. So for our UPEC mutants, 4 out of 10 of the mutants were confirmed to be fully phage resistant. 4 out of 10 of the mutants were found to be partially phage resistant. And then 2 of the 10 mutants were actually found to be fully phage sensitive. They had no resistance. And so here we found that antibiotic sensitivity varies by the mutant, and that depends on the level of phage resistance. So here's another figure showing the efficiency of plaquing. Again, the 10 mutants are on the x-axis and the efficiency of plaquing is on the y-axis. So for the 2 mutants that had no phage resistance, you can see that there's no real change from the wild type bacteria. In the 4, which had partial phage resistance, they have varying levels of partial phage resistance. Specifically, this value of Z indicates that we saw zones of lysis, but never formed individual plaques. So we weren't able to quantify the efficiency of plating. And I'd also like to call attention to this strain, AB376, which out of these 4 is the closest to complete phage resistance. And then for these 4, as shown in the previous figure, they're all below the limit of detection. And so those are completely phage resistant. So moving on to the antibiotic sensitivity results, for these 2 strains, which showed no phage resistance, these values are around 1, meaning that there was no change from the wild type bacteria. And then in the 4 with partial phage resistance, these varied a little bit more, but we don't see very many trade-offs in the antibiotics. However, in AB376, which I just mentioned is the closest to complete phage resistance, we do see more sensitivity in the antibiotics as compared to the other partially phage resistant mutants. And then finally, in the 4 mutants that we found with complete phage resistance, you can see across the board for all 6 antibiotics that there is a trade-off here between phage resistance and antibiotic sensitivity. So in conclusion, we found that host genetic background does influence the relationship between resistance to phage U136B and sensitivity to antibiotics. We found that in UPEC mutants, antibiotic sensitivity depends on partial or complete resistance to phage U136B. And this preliminary trade-off between complete phage resistance and antibiotic sensitivity in UPEC makes phage U136B a potential candidate for phage therapy that restores antibiotic sensitivity. So in the future, I'm currently isolating and testing more phage-resistant mutants from each strain for a total of 20 from each strain. Also currently measuring MIC for more antibiotics, including colistin and tetracycline, which Alita talked about in her presentation. We also have plans to do bacterial genome sequencing to infer the mutations that are conferring phage resistance. And finally, in the future, we'd like to conduct evolution experiments for phage U136B in the presence of E. coli B strain and UPEC, like has been done with E. coli K12 strain. So I would like to acknowledge all the members of the Turner Lab and the following funding sources for the support and help that they've provided for this research project. And I would love to answer any questions.
And again, anyone can jump in with questions or we can read them from the chat. So a question from Ben Kerr. I'm curious if the reverse experiment selection for drug resistance and measuring effects on phage resistance has also been done. What do you think are the prospects for cocktail phage plus drug therapy from this work?
So we have not done those reverse experiments. I think that could be an interesting future step also. As far as the phage cocktail ramifications, I think that this is an interesting study in obviously complete versus total phage resistance. And so we haven't specifically started thinking about maybe testing that with phage cocktails, but obviously that's something that's super relevant to the application of phage therapy. And so I think that this could also shed some light on the mechanisms going on there. So I think that would be some work that would be really cool to do in the future as well.
Kyle, you had a question earlier about partial and complete phage resistance. Do you want to expand on that more?
Sure. Could you remind me of the question?
Oh, sorry. I meant Kyle, if he's still around at the moment, had kind of asked about looking at partial or complete phage resistance in these, or even in the other BW25113 mutants, which I think you had also looked at.
It looks like Kyle might have had to disappear.
Oh, sorry, I just returned. What was the question?
Oh, we were just doing open Q&A, but earlier you could ask a question about partial or complete resistance.
Oh, yes. Yeah, what does that correlate with trade-ups or trade-offs? Yeah. But yeah, cool work.
Thank you.
I have a question / suggestion. So great job, Carli. At least in Pseudomonas, and I don't know if this is true for E. coli, but efflux pumps are upregulated in biofilms. And so I think they play a bigger role in antibiotic resistance in the biofilm state. So have you guys ever thought to look at biofilms and see what your antibiotic resistance phenotype might be?
So we haven't thought about that for this specific project. I know there are some folks doing some of that research in this lab, but we have yet to sort of think about how that might play into this project. But I also think that that would be something really cool to look into, and again, important for potential applications of phage therapy.
Okay, great, thanks.
If it's allowed, I'd like to ask a question of Chris, who asked the last question. I'm wondering what the logic, whether there's an obvious explanation for why the efflux would be upregulated in a biofilm. Is it just that the collective power of all that efflux kind of might work better in a biofilm? And yet it also seems like, well, you're just pumping an antibiotic into the environment, which is where your nearby neighbors are. So it also seems like it might be counterproductive. So I'm just wondering, has anybody sort of looked at the fitness consequences in a biofilm of differential regulation of efflux?
Well, this is speculation here, but I would speculate it has nothing.
Chris, you're out of time.
Oh, all right.
but maybe we can take that to the chat. But yeah, great question. I'm curious about that answer as well.
Hello, everyone. I want to apologize in advance that my camera is acting up. I do have a video on a separate device if you are curious as to looking at my face while I present, but I'll continue here on my laptop. So I may be looking in two different directions. So my research is on the assessment of the evolution of Streptococcus mutans biofilms in microgravity. On the picture there, that's me, and I go to A&T. I'm a second-year master's student. So, what are biofilms? In layman's terms, they're the aggregation of free-flowing planktonic cells. Once these cells adhere to a support solid surface that they like, they mature and make a mature biofilm. S. mutans, the bacteria that I study, is an etiological agent of caries. It's gram-positive and a facultative anaerobe. You can see here on the bottom picture the free-flowing planktonic cells that I mentioned on the left and how it can form into a biofilm on the right. Dental caries is just an array of dental diseases that form because this S. mutans allows other bacteria to adhere to the surface of the tooth, causing other dental issues because of the low pH and demineralization of that enamel as these cells adhere. So I'm studying microgravity because astronauts often display a decreased ability to fight off bacteria in the mouth because of the decreased salivary lysozyme. And with this, you have increased dental calculus, causing things like periodontitis, gingivitis, and cavities. That yellow film around the gums is an example of how that biofilm can disrupt the health of the tooth. NASA is interested in understanding these issues with dental caries because the long-term physiological response to S. mutans hasn't been thoroughly observed. They predict that it'll be one of the top 5 medical conditions negatively impacting future space missions. The studies that we have now have 1 to 2 samples of S. mutans in microgravity, but with 24 to 72 hour time ranges. So the long-term changes with S mutans and biofilms hasn't been thoroughly observed. And it's especially relevant because missions typically last 4 to 6 months. So with my experiment, as you know from my colleague Mizpha, we use high aspect rotating vessels or HARVs that put the bacteria in a microgravitational state. So they rotate and decrease the gravitational vectors acting on the cells. I believe, if I'm not mistaken here, the normal gravity conditions on Earth are 9.8 to 9.9 meters per second squared, whereas in these HARVs, the cells experience about 0.5 meters per second squared gravity forces. And so our normal experimental evolution protocol calls for an ancestral population of S mutans to be split between microgravity and normal gravity populations. We maintain these cultures for 100 days. We store back up glycerol stocks every few days, twice a week. We subculture 36 to 48 hours for 100 days to maintain the cells in the HARVs and do phenotypic assays to assess changes in adherence, acid tolerance, oxidative stress, and antibiotic resistance. At the end, we do a whole genome-wide assessment of the bacteria over the 100 days to see what kind of genetic changes have occurred. And so our previous microgravity study between microgravity and normal gravity showed that there was some changes in acid tolerance that may be associated with microgravity, but nothing in adhesion ability or antibiotic resistance was specific to microgravity. And this was just done with the planktonic S mutant cells. Something interesting that we did find in our second experiment with the experimental evolution protocol with S mutans is that between a microgravity population and a microgravity population exposed to silver over the course of 100 days, there is an increase in antibiotic resistance associated with increase in silver resistance. So on this chart, you can see that in the green and purple, that's day 55 for 2 microgravity cultures. The minimum inhibitory concentration of silver is about 12 micrograms per mil at day 55 versus day 88 at green and red, at blue and red, excuse me. You have the minimum inhibitory concentration at about 17 micrograms per mil. And so that was interesting to see that if exposed to silver long-term, these bacteria can develop resistance as expected. Also, some of the phenotypical observations we observed just from maintaining the cultures, you can see on the right that the colonies actually change. Initially, we thought between those 2 colonies, you would assume contamination, but those are both S mutans cultures. One is more lustrous and round, the other one's more flat and dull. And my research objective is to determine the effect microgravity has on the evolution of biofilms, because we believe that the virulence and changes in virulence in S mutans may be reliant on the formation of biofilms, where they can develop more resistance and more protection from stress, environmental stressors. And so I want to include hydroxyapatite into my HARVs over 100 days to assess the biofilm growth. And hydroxyapatite, if you don't already know, is that mineral that forms the enamel of the tooth, the human tooth, and it's responsible for repairing the enamel. So any kind of microfissures or demineralization that occurs otherwise is repaired using hydroxyapatite. And it's currently used for other dental disease studies where they adhere hydroxyapatite to a surface to assess biofilm formations. And so to revisit that experimental evolution protocol that you may have seen in my colleague's presentation, we take the ancestral S mutans and just add it to a HARV that has hydroxyapatite disc stuck to it. We do the same thing with a microgravity and normal gravity populations where the discs are adhered to the surface. And we continue with the contamination test and store stocks for future experiments twice a week. We subculture every 36 to 48 hours and do the phenotypic assays as normal. The only difference is I'm assessing adherence and acid tolerance with my biofilms versus oxidative stress and antibiotic resistance. And at the end, we also do the whole genome-wide assessment of the planktonic, the biofilm cultures. And there you can see that little white disc is an actual hydroxyapatite disc that's stuck to the HARV, and the beads that I use here are also hydroxyapatite. They assist with my phenotypic assays, where we can gather a snapshot of what the biofilms are doing at these 3-week time points over 100 days. And so currently I only have 3-week data, but I basically have done a few phenotypic assays with this 3-week assessment. This first one is acid tolerance, wherein I see how many cells are alive versus how many are dead after being exposed to acid for 0, 20, and 30 minutes. There's no trend between the normal gravity NG and microgravity MG populations, but this is just 3 weeks out of 100 days. And then after 24 hours with the introduction of the hydroxyapatite beads, you can see that there's more adherence between normal gravity versus lower adherence in microgravity populations. These are both quantified using green-red fluorescence ratio readings. And we can see basically how many cells are alive versus how many are dead between the normal gravity and microgravity populations. In the future, I want to incorporate GFP plasmids into my bacteria for enhanced quantification methods, especially using those hydroxyapatite discs. I also want to develop efficient ways to assess oxidative stress and antibiotic resistance, just to determine more physiological characteristics or changes that occur over these 100 days. And the use of scanning electron microscopy would also be efficient for assessing these cells as they are maintained in the HARVs. And lastly, I'd like to thank Dr. Misty Thomas and the Thomas Lab, and also my sources of funding, which are North Carolina Space Grant, the Triangle Center for Evolutionary Medicine, and my NIH BTD Fellowship, Bridge to the Doctorate Fellowship, headed by Dr. Martin. Thank you.
Great, thank you. And we have plenty of time for questions. So I have a question, but first I just want to say that I think these experiments in the microgravity chambers that you and Mizpha have been working on are just so cool. It sounds like a really challenging experimental setup, and you only have so many to work with and dealing with contamination and everything. So it's really, really cool to see that. My question is, when you had the hydroxyapatite in the chambers, were those pieces of a certain size, or was it sort of at a very micro scale?
The pieces of hydroxyapatite beads?
Yes.
Oh, actually, I'll try to go back to make sure I understand. So you're talking about those little beads at the bottom, right?
Yeah, yeah.
Yes.
Okay, so it's quite small, kind of like a powder.
Right, it is a powder. They're micron, just links. I'm not sure the diameter exactly, but they're very small and can be seen under microscope. They're about a little bit bigger than the size of my S-butan cell. So.
Okay.
I guess this is Nkrumah at the University of Idaho. First and foremost, you had an amazing talk. So I guess since we're talking about the hydroxyapatite, so obviously you're setting up some structure in your chambers and then you add them. And I think cells on the outermost periphery of the device then are going to experience more or less microgravity than those in the center. Have you thought or considered, changing the location of those discs, right, to reflect that?
Actually, I have. So this is preliminary data. This is really a new experiment for me. This is actually the first time that I've gotten these cells in the HARV. So right now I'm really optimizing the way that I'm going to do this experimental evolution protocol. Thus far, it's been sort of successful, but again, there's always room for improvement. And that would include changing those hydroxyapatite discs to maybe the sides or having both the sides and the center. But I'm kind of limited based on the size of the HARV. So the volume, I mean, there's so much to work with in 4 mil volume, but again, I have considered changes.
Awesome, great experimental setup. I agree, this is very, very cool work.
Thanks.
Thank you, Leah, for moderating the session.
Happy to do so. So thanks everyone for attending. We can stick around for a while, any of the speakers who might want to stay and chat. Otherwise, great job to all of the speakers. Thank you for contributing to this session.
Yeah, thanks everyone.